Okay, welcome, my dear friends. Regarding of our free online coaching for all competitive exams related to plant sciences, plant biotechnology for CPGET 2025, CUET, and other competitive exams like Forest Department, Horticulture and Agriculture Department. So today's our eminent resource person, Professor E.M.Sunitha Madam Garu, working as Head Department of Botany, one of the prestigious institution in Hyderabad. Babu Jagjeevan Ram Degree College, Autonomous, Narayanguda, Hyderabad. So, Madam has used experience in different colleges, and also Madam has very friendly and very cooperative. So, in this regard, we are associated to organizing this type of free online coaching for our young budding botanists and also the young aspirant, those who are going to attend CPGET 2025 and other competitive exams. So, our Professor E.M.Sunitha Madam Garu is rigorously joining for us. So, Madam, please welcome and start your session. Please welcome it.
Thank you so much, sir, for your kind words. Good evening, all my dear students. Welcome to this coaching. Actually, I thank Dr. Tirupati sir for taking this good initiative and starting this free online coaching. Today, this is the first year we are getting associated with you. Hope this association and collaboration will continue for years. And with this, I would like to start my topic today. Students, today I'll be dealing with plant tissue culture. So first, we must know what is plant tissue culture. That is culturing something from the tissue in the plant system. Okay. So what we in this class we will be knowing what is the basic difference between plant tissue culture, animal tissue culture, what are the different types of tissues we can use for culturing the plants, and so many other things, and why we are able to do plant tissue culture so successfully than the animal tissue culture. Okay. So when we come to the definition of this plant tissue culture, first we must remember that plants has a property called as totipotency. Okay. So plants have a potential called as totipotency. Because of that particular potential, we could be able to, or the scientists could able to do or start the tissue culture of plants. So when we come to the definition of this plant tissue culture, it says that we can culture a whole plant using the seeds or organs or tissues or cells or protoplast on a nutrient medium, that is artificial nutrient medium under sterile conditions. And what these people are telling that whatever the plant material is there, the whole plant can be used as a explant from which you can do the culturing of the whole plant, but under certain conditions. What they are telling under certain conditions, they are saying those conditions as within nutrient media, like how animal system and plant system, we require food for our survival. In the same way, because we are doing this under in vitro conditions, ante in vitro conditions means under laboratory conditions. Ante manamu eppudaithe baita outside when we are doing dani manamu in vitro conditions అనేసి అంటాం అన్నమాట. ఎప్పుడైతే ఒక లివింగ్ సెల్ లేకపోతే ఒక లివింగ్ సిస్టమ్ లోపల డైరెక్ట్ గా చేస్తారో, యు కాల్ దట్ as in vivo conditions. So tissue culture మనం ఎక్కడ చేస్తున్నాం, how where we are doing it under the artificial or outside or under the laboratory conditions. So దాన్ని మనం you call that as in vitro conditions. So in vitro conditions లో what it requires nutrients. So naturally the nutrient medium has to be present and it has to be microorganism deficient. That means any microorganism should not be present there. So a sterile condition has to be present. Why? Contamination chances are more. So what the scientist has given, what scientists who have done work on tissue culture, what they have given the definition is, you can culture the plant from seeds or from the organs or from tissues or from cells or from even from the protoplast also, where in the nutrient media under sterile conditions. So that's a definition of this tissue culture, from where how we can do it. But before we go to the tissue culture, what is the procedure and all, we should see it is we have to learn some basic things in this tissue culture. What are the basic things? Is we are telling, yes, nutrient media is enough, but how we have hormone system, we have the endocrine and the exocrine hormone system in our body. In the same way, plants also, it has certain hormones called as the phytohormones, or the growth hormones are present in the plant system also. Simply just giving them the micronutrients, macronutrients, sucrose, it's not enough. It definitely requires certain growth hormones. Those are very, very basics of the tissue culture. So if we see this, if we study this, only two different hormones are enough, or two different hormones play important role in this development of this tissue culture from whatever explant you are taking. What are those two hormones? Those two hormones are auxins and cytokinins. So auxins, what they do, they stimulate the root development, and cytokinins, what they do, they stimulate the shoot system. Okay, so these two hormones has to be given for both. If you are doing root development, both auxins and cytokinins has to be given. If you are doing shoot development, then also you have to give auxins and cytokinins. But there is a ratio. We have to remember that ratio. So what is that ratio is, when we are going for the root development, the auxins concentration should be more when compared to the cytokinin. Because what cytokinins will do? Cytokinins will help in the cell division and in the cell wall formation. It will help in the cell division mostly. So definitely that is required. But in the root development, the auxins concentration should be more when compared to the cytokinin. And it is vice versa. Ante shoot development lo, you require cytokinins more and auxin concentration less. But in between this shoot and root development and the explant, you have one more stage called as the callus development. In that callus development, you should have equal proportions of auxins and cytokinins. Okay, so when we are doing under the laboratory conditions, when you're taking some explant, that means any type of plant, let it be rice, or let it be tobacco, let it be carrot, let it be any dicot or monocot plant, when you take, first you have to standardize the procedure. Ante dani manamu, if you are going some ఒక కొత్త ప్లేస్ కి పోయినాం, వాట్ వి సే, మనం అడ్జస్ట్ కావాలి ఆ ప్లేస్. సేమ్ లైక్ దిస్, ఇఫ్ యూ వాంట్ డూ అండర్ ద laboratory conditions, మనం రీసెర్చ్ కి వెళ్తున్నప్పుడు స్టాండర్డైజేషన్స్ ఆర్ వెరీ వెరీ ఇంపార్టెంట్ అనమాట. సో అట్ దట్ టైమ్ callus development will be very very easy for us, because both auxins and cytokinins మనకి సేమ్ కాన్సన్ట్రేషన్స్ లో ఉంటుంది కాబట్టి. బట్ you require lot and lot, I think 1 year or 1.5 year of time will take for the root development and shoot development protocol or standardizations cheyadaniki. So auxins and cytokinins, they play an important role in the research field. How the plants, just we are seeing in the nurseries, so many of plants, tomato plants, you are having chilies, so many broccoli, so many tissue culture plants are there. But how they have developed, we are just studying, yes, cytokinins and auxins like that, but so much of standardizations procedures are required. That's why you should remember, in the questions they will ask, what is required more in concentration, auxins or cytokinins for root development? Or otherwise they can just give you a random question. So and so a plant was being tissue cultured, the shoot has already developed, but the root has to be developed. Which hormone has to be given in more concentration, which hormone has to be given in less concentration? So that we have to see and we have to write the question. Okay, now next, after we have studied that auxins and cytokinins are very important, next we will go see, we will see here how we control the in vitro culturing. So we can control the plants, adventitious roots or shoots or callus, all this can be controlled stage by stage. So what we will have, first if you are taking some root strip as X plant, so first you will be having the auxins. Next, from there you will be getting the shoot system. Then you will be getting the root system. Okay. So when you increase or decrease the cytokinins, with that you will be able to do the thing. So here in the previous slide, as we have discussed, that auxins is required more for the root development, and for the shoot development, cytokinins are required in more amounts or more concentrations it is required. Okay, that is very, very important. So when we see the tissue culture just in a glance, what we find here is you're having the plant here, the whole plant you're having here. So this you call that as a mother plant. From this, you have to take some mother culture you have to take. So you can take anything, as I have discussed in the first slide. You can take the seeds, you can take the leaf, you can take the meristems, you can take the protoplast, you can take tissue, or you can take any part you can take. And you have to put it in the nutrient media. In this nutrient media, you will be having the same concentration, equal concentration of auxins and cytokinins. Okay. మనము ఫస్ట్ ఎప్పుడైతే మదర్ కల్చర్ తీసుకుంటామో దాన్ని మనము క్యాలస్ ఫార్మేషన్ చేసేటప్పుడు బోత్ ద కాన్సన్ట్రేషన్స్ విల్ బి సేమ్. సో మనకి క్యాలస్ అన్నది వస్తుంది అనమాట. క్యాలస్ వచ్చిన తర్వాత ఆ క్యాలస్ ని మనము యూ డూ ద ప్లాంట్ రీజనరేషన్. దానికి ఫస్ట్ షూట్ డెవలప్మెంట్, దాని తర్వాత నెక్స్ట్ రూట్ డెవలప్మెంట్ అవుతుంది. దాని తర్వాత again you have to take that and you have to transplant into the sterile condition pot in the vermiculite అట్లాంటిది. Only after that you will be taking it into the outside condition మనం తీసుకొని వెళ్ళచ్చు. దిస్ ఇస్ జస్ట్ అ గ్లాన్స్ అనమాట. ఇది జస్ట్ ఇట్లా మనకి గ్లాన్స్ లాగా చెప్తున్నాను. బట్ యాక్చువల్ థింగ్ ఇస్ మోర్ what we see in this slide. Okay. So now coming here, first we have to see, if you are doing tissue culture, in that lot and lot of limitations are there. So what are those limitations, or what are those factors manaki manchiga tissue culture plants ravadaniki or manaki worst avvadaniki? You have to have considered so many factors anamata. So what are those factors? First and foremost factor is the growth media. As I told you, we are dependent on food anamata. We have balanced food. What type of balanced food we have? We have the carbohydrates, we have the sugars, we have lipids, we have fats, and minerals and vitamins. Same like that for the tissue culture also, for the plants also, they require growth media. A growth media lo em untundi, it will have minerals, it will have growth factors, it will have carbon source, and